Welcome back to the biosignaling playlist, and in this video what we're going to do is we're going to discuss the G protein. So we're going to discuss G proteins. And G proteins get their name from the fact that they bind GTP. So G proteins are able to bind GTP, right? And so what you have is typically in textbooks they'll use one type of G protein system just to illustrate the function of all of them. And so that's what we're going to do in this video. We're specifically going to look at the adrenergic G protein system. So we're going to look at the adrenergic G protein system. So let's get familiar with what we have here. So what we're going to have is we're going to have the adrenergic receptor. So it's going to look like this. So here's my adrenergic receptor. And the receptors in G protein systems, they are called heptahelical receptors. They're heptahelical. And what this essentially means is that you have the protein sort of snaking through the membrane 7 times. So what you have is something that looks like this. So you have the N-terminus out here, and then it's just going to snake through 7 times. So here's 1, 2, 3, 4, 5, 6, and then it's going to kind of snake back here, and then here's the C-terminus. And this is your adrenergic receptor, right? Also over here we have a G protein, right? So the G protein is actually divided into 3 functional areas. The first part, these two sort of go together. So I'll kind of do it like this. And over here you have the beta subunit, and you also have a gamma subunit. These parts, what their function is regulatory. When the alpha subunit is bound to them, the alpha subunit's inactive. So over here you have the alpha subunit, right? And the alpha subunit specifically is the subunit that binds GTP. And when GTP is bound to the alpha subunit and epinephrine is bound to the receptor, the G protein becomes active, specifically the alpha subunit. Okay. Now over here, let me do this in purple. We have another enzyme, and this enzyme is called adenylate cyclase. So this enzyme is called adenylate cyclase. And what this enzyme effectively does is it takes an ATP molecule, right, and it turns it into basically 3', 5' cyclic AMP, and of course there's a loss of pyrophosphate in the reaction. So actually, real quick, let's actually take a look at what cyclic AMP looks like, just so we can get a sense of what's happening. So of course it has the characteristic ribose ring. Okay, it has the ribose ring. Up here it has the adenine nitrogenous base, right? Of course that ring is aromatic. Let me fill that in. It's aromatic. It's got its amine up here. And then it sort of cycles back like this to form the ring. So that's why they call it cyclic AMP, because it's cyclic, and it also has 1 phosphate, so cyclic AMP. So whenever, you can imagine when epinephrine is bound to the receptor, and of course when the alpha subunit has bound GTP, right, you could imagine that of course there's going to be activation of adenylate cyclase, and you're going to have a lot of cyclic AMP. So let's assume that there is epinephrine present. So epinephrine is going to look like this. Epinephrine is going to look like this. So I've got my amine over here, my hydroxyl groups. There's epinephrine, and it's going to interact with the adrenergic receptor, right? And when it interacts, it's going to cause a conformational change in the receptor. And that conformational change translates to the G protein. Now, one thing that's important to understand is the alpha subunit will only dissociate from the rest of the G protein if GTP is bound. So there's sort of a constraint on the activation of adenylate cyclase. You have to have epinephrine bound in the receptor, and the alpha subunit has to have bound GTP. If both of these things are the case, then the G protein will sort of dislocate from the other subunits of the G protein, and it will move along the membrane, and it will reach adenylate cyclase. So it'll sort of, I'll draw it kind of in parentheses or whatever here. And so here's the alpha subunit, and of course it still has bound GTP. So here's the GTP, right? And when it binds to adenylate cyclase, it activates it. And when adenylate cyclase gets activated, of course you have the catalysis occurs. ATP gets hydrolyzed, and you end up cyclizing it to cyclic AMP, and you have the loss of pyrophosphate. And actually, one other thing that helps drive the reaction is the inorganic pyrophosphatase. And of course, that hydrolyzes pyrophosphate into 2 inorganic phosphates, right? But anyways, so you can imagine if there's lots of epinephrine present and GTP is bound to the G protein alpha subunit, you could imagine that there's going to be a lot of cyclic AMP. So initially, when this happens, there will be a lot of cyclic AMP. As we'll look at in later videos, there's actually ways to terminate the response, but initially there is a lot of cyclic AMP. Now, there's another protein that cyclic AMP activates, and this is going to test my artistic skills, but anyways, what it's going to look like is essentially this. So it has this protein right here, and this is going to be a protein called APAC, and this stands for excuse me, not APAC, it's ACAP. ACAP, sorry. It's ACAP, and this stands for protein kinase A anchoring protein. So it's an A kinase, a class A kinase anchoring protein, and it's bound to another protein. I'll do this in orange. It's bound to another protein, and this protein is a regulatory subunit. Okay, and as we'll find, it regulates the activity of protein kinase A. So I'll do protein kinase A in red. Here's protein kinase A in red, right? So here is protein kinase A, and each one of these anchoring proteins contains 2 protein kinase A's, right? Now, the protein that's in orange, so the protein right here that's in orange, right? This is a regulatory subunit. And when the regulatory subunit is bound to protein kinase A is held inactive. It's held inactive. But it turns out that when cyclic AMP accumulates, the cyclic AMP can actually interact with the regulatory subunits, and that causes dissociation of the protein kinase A. So this is how cyclic AMP activates protein kinase A. It binds to the regulatory subunit that links the anchoring protein to protein kinase A, and it causes dissociation of protein kinase A. So ultimately, when cyclic AMP levels rise, you have an increase in the activity of protein kinase A. Well, the point that we have to illustrate with protein kinase A is it initiates a cascade of phosphorylations. Okay? Now, in the context of epinephrine metabolism or epinephrine biosignaling, protein kinase A is going to activate another protein, and it's called phosphorylase B kinase. Okay? Phosphorylase B kinase. And so as you increase the amount of protein kinase A, you increase the amount of phosphorylated phosphorylase B kinase. And when this molecule gets phosphorylated by protein kinase A, it activates another enzyme. And this enzyme is called glycogen phosphorylase. And if you've watched the glycolysis playlist, you know what this enzyme does. It's essentially going to take a glycogen chain, right, and it's going to phosphorylate it specifically at the 1 position of the non-reducing end, and it's going to generate glucose 1-phosphate. And the glucose 1-phosphate can be isomerized into its constitutional isomer glucose 6-phosphate, and that can go into glycolysis. But that's of course the case for epinephrine, but the main point with respect to G protein systems is that the protein kinase that's activated by the increase in cyclic AMP is going to induce a phosphorylation cascade. And then the phosphorylation cascade is ultimately what induces the cellular effect. So what did we see? Well, we saw that the hormone epinephrine bound to its adrenergic receptor and induced a conformational change that when in the presence of GTP, the alpha subunit changes conformation and dissociates from the rest of the G protein. And of course, it activates adenylate cyclase, and the levels of cyclic AMP increase in the cell. And when cyclic AMP binds to the regulatory protein that's attached to the anchoring protein, it causes dissociation of protein kinase A. And of course, then the rest of the cascade we looked at. So ultimately what epinephrine does is it increases the amount of glucose 6-phosphate in the cell. And ultimately that's used for energy in glycolysis. So I hope this video helped you give you a little bit of an intuition on what's happening in G proteins. In the next few videos, we'll actually look at some more detail on it and also how the effect is terminated. See you soon.